Brazil. What I'll be talking today is about biomarkers and tinnitus, which is a quite challenging area. What we know about tinnitus, so the ringing in the ears, that's a phantom sensation that maybe some of you, the young people, have experienced when they go to a discotheque or party and you have a buzz in the ear that maybe lasts for overnight, and then your hearing recovers. But for some people, it's very, very, very annoying, and it can be all the time present at very loud perceived levels. And for these people, there is no cure for this. And we think that one of the reasons for this lack of treatment is because tinnitus is very heterogeneous. And we don't know yet what are the subtypes. Subtypes could be like a subgroup of defined and homogeneous phenotypes. We know, for instance, that there are some tinnitus that are objective. That means that the ear produces a sound that can be measured outside. But the majority of them is subjective. That means it's only the person that is living with this perception that has this phenomenon. Some of the tinnitus can be pulsatile, so it can follow heartbeats, or it can be also sensed like a Morse code, like that. And for some other people, it's non-pulsatile. And we've seen that the pulsatile ones are really much more impacting on life quality. And then if you have a tinnitus that is more like of a sound, it can be intermittent. That means you can hear it now and then. It can be occasional, but it can also be perceived constantly. And there, what makes it kind of complex is that you can have a lot of different etiologies to it. You can have hearing loss, whether it's conductive or sensorineural. You can have vertigo effects combined with Meniere's disease. You can have headaches, psychiatric conditions like stress, anxiety, depression, but also somatosensory problems. It's been more and more known that the neck stiffness and also the temporomandibular joint issues can contribute to tinnitus. Now, so we see here a multidimensional scale of tinnitus, but this also encompasses severity, which is typically measured with questionnaires like the Tinnitus Handicap Inventory, Tinnitus Functional Index, which measure the stress level, and then the time component, which can be acute or it can be long-lasting and chronic when it's more than a year. So with all of this, we believe these could be subtypes, but there is no evidence for this really. So one of the first things we started doing in the laboratory is to first ask questions about how the genes and the environment together interact to generate tinnitus. If you consider yourself and your phenotype, you are a combination of genes and their interaction with the environment. If you consider here this graph, it shows that monogenic diseases contribute to 100% to the phenotype. And the opposite here, you have environmental factors like injuries, nutrition, and these would contribute totally to the phenotype here. And in between, we have a combination of both. And in the genetic epidemiology, there's been always this question of how much genetic can contribute to a trait when compared to the environmental. So what we've done at first, we took advantage to some of the available data in Sweden. Sweden is well known for their studies involving people from the public. And we did those studies in twins, in which we've been asking, if twins are identical, what is the likelihood, if one twin has tinnitus, what is the likelihood that the other one has it too? And there we saw that for bilateral tinnitus, a tinnitus that is perceived on both ears, there is a heritability of 67%. 100% meaning the maximum genetic contribution. And this was a significant contribution of tinnitus. Another way to address this is to check with the adoptees. So if you have a child that has tinnitus and you can verify whether the adoptive parents also have tinnitus, you can answer the question whether the shared environment has an influence on the generation of tinnitus. And for this, we've been using data from the whole nation in Sweden. So the Swedish government has a database where you can track individuals for research purpose. And we identified adoptees that were diagnosed with tinnitus, and we could track their adoptive parents. and also verify whether they had tinnitus. And then we went back and looked at the biological parents, and there also we verified whether they had tinnitus. And the message here is that if your adoptive parent has tinnitus, you as a child had no more greater risk for having tinnitus. But if your biological parents had tinnitus, then there was a significant contribution from that genetic environment. And here the heritability was calculated as 31%. And there we went further into doing a familiar aggregation study with our cohort, where we found nearly 3,200 relatives from which we could inquire, if one person had tinnitus reported, what is the likelihood that another sibling also had tinnitus? And there we could stratify depending on different forms of tinnitus, whether it was bilateral, unilateral, constant, occasional, or severe. And there, severe tinnitus came up as a very high trait that was influenced by genetic transmission within the family, with the likelihood from 5 to 10 of another sibling being also impacted with tinnitus within the family. So what this kind of told us is that depending on the subtype, so for instance here, this is bilateral tinnitus. Here we have significant tinnitus, so meaning diagnosed by a doctor. And here we have severe tinnitus. Depending on that, we would have different influence of genetics into the liability to obtain tinnitus. For this, the genetic evidence that tinnitus could be in the genes came from the very first large genome-wide association study that was published by Clifford et al., so a group in San Diego. And they found out using the UK Biobank, which had nearly 200,000 people, that they had a number of loci here found significant and replicated with another cohort, which was the Million Veteran Program. So this was the real first time that some genetic regions were found associated with tinnitus and could be replicated in another independent cohort. And there they could perform some genetic relationships with other traits, and they found a strong correlation with major depression disorder. So this first work was very interesting because it set the basis for future work. And in Sweden, we started creating EarGen, a consortium to dig on the genetic contributions to hearing loss and tinnitus. And Natalia, who's a PhD student in the lab, she comes from Macedonia, started reasoning, and she said, all right, if we have now a number of cases, other studies have shown that if we increase them, then the likelihood of finding more single nucleotide polymorphisms would also increase linearly. So we started digging another consortium with different collaborators, the Swedish Twin Registry, the Estonian Biobank, FinGen, which is in Finland, and the Danish Twin Registry. So we had like a Scandinavian combination of cohorts. And at the same time, we figured out that other colleagues were also working on the same project, Nachtigall and Williams, and they had another number of cohorts from Europe and the US. And combining efforts together, we reached a sample size that was decent enough to start to ask the questions, what is the genetic factors that would be contributing to hearing loss and tinnitus? So if you look at tinnitus here, it was quite disappointing to see that we had very few genetic loci being significant. This one and this one here. So 2 loci, 5 mapped genes, 2 lead SNPs. This was not a lot. And when we compared it to hearing loss, you see here that we have the majority of the signals coming here, 39 genomic loci. This is a manuscript that is now in preparation. So 142 mapped genes. This was really a big difference. And we started asking ourselves what was the problem here. But before we go back into that issue, I want to share some of the findings we had for the hearing loss study. Here we had self-reported hearing loss, and there's been studies that have found a very good correlation with self-reports in hearing problems, but also the pure tone audiometry. Here we believe that tinnitus could have been phenotype so differently among the different cohorts that this has triggered a lot of noise, and the signals have kind of disappeared in this mess of blurry definitions. For hearing loss, we took advantage of single-cell RNA sequencing data that was collected by Decibel Therapeutics. So that's a pharma company that works on hearing. They had a number of cells from the spiral ganglion neurons and also the stria vascularis that you can see here. And we obtained different data sets also from the organ of Corti. This is mentioned here. And the Karolinska also had a number of data collected from the brain, and you can see here all the different regions that summed up to 160,000 cells. And what was interesting to see is that while there was no enrichment of any of the GWAS-SNP signals in the brain, we could detect some signals, for instance, in the stria. So here the main question was, what is the cell type mainly responsible for hearing loss? In contrast to what we expected, we found no enrichment in outer hair cells and inner hair cells. This was not significant. Instead, when you look at the other cells here, we see that the spindle and the root cells and the basal cells from the stria vascularis were significantly enriched with SNP signals from humans with hearing loss, which suggests that these cells would be the major contributors to age-related type of impairment, in contrast to the well-known role of the outer hair cells and the inner hair cells in hearing. So one of the ideas now is to perform the same type of analysis with tinnitus, but we need to really solve the problems of definition across the data sets. So for that, understanding how we should subtype tinnitus and stratify it, we had our project in Sweden called STOP, the Swedish Tinnitus Outreach Project. And one of the goals of that project was to try to define subgroups based on electrophysiology or blood biomarkers, but also genetic variants. And here you have the different contributions that have emerged so far from those studies. And I will go briefly throughout some of these so you can understand the cohort and what it's been made of. One of the pipelines that we've been using for this is to start collaborating with different cohorts locally. They had data on tinnitus, so the Swedish Tween Registry, which we showed you the data with tweens. SLOSH, which I'll show you data right afterwards. It's a longitudinal occupational survey of health that has very interesting data on tinnitus as well. LiveGene, who has DNA here, and all their participants joined STOP, and I'll show you more data afterwards, the Stockholm Public Health cohort, and then we had also some public outreach to try to gather some additional participants. And here the strategy was to start surveying them with questionnaire data, and then to invite a number of people that were locally in the Stockholm region and to invite them for some audiometry testing. And then with this, try to fiddle down and obtain homogeneous groups that we could analyze and test for genomics, proteomics, and electrophysiology. Sorry. So here, the SLOSH cohort, we were able to start asking the question about the dynamics of tinnitus. So we had data collected from 2006 up to 2016, every 2 years. And there the question is whether you had tinnitus, and the answers were no, sometimes, often, or always. So sometimes or often are categorized as occasional tinnitus. And what we notice is that these people would transition between groups quite dynamically. And surprisingly, the more frequent tinnitus were perceived, then the greater likelihood that it would remain constant throughout the time was greater. And we made a calculation for this. You can see here that if you perceive tinnitus sometimes, the odds to get it constant 2 years later was around 5. If you had it often, this rose to 31. And then if you had it constant, this was even higher, up to 403. And so this, what it told us is that eventually, as tinnitus is transiting to a constant perception, something must happen here that freezes tinnitus and makes it less likely to go back into the other groups there. So there would be a change in the physiology. So to answer that question, another PhD student, Nicholas, has been working on this project for over 5 years, and this recently came out. He surveyed participants first, and we had a screen then to get individuals invited in the clinics, two in Stockholm and one in Lund. We excluded a number of participants for the reasons that are present here, and then we divided the people in different groups, those that didn't have tinnitus, those that had occasional tinnitus, and another group that had constant tinnitus. We started collecting data with a clinical system called Charter from Interacoustics, but you will see afterwards that some of the wave responses from the auditory brainstem recordings appeared to us not equal to what is typically expected. And then we tested another system too, which is why we have the two of them here. But we made the statistics in order to take these aspects into consideration. So you can see here the pure tone audiogram. So that's the level of hearing that is present. Here, the line is the threshold of hearing that is determined by the WHO standards. So anything that is below here is defined as some degree of hearing loss. So if you measure from here to here, this is roughly 25 dB in hearing deficits. And you can see here that the degree of hearing, the no tinnitus, the controls are in green, the occasional are in blue, and the constant are in red. And you can see that there is the more constant the tinnitus becomes and the more hearing loss you have. There you have the left ears and the right ears. You can see the patterns are alike. And here are loudness discomfort levels, which is a measure of sound tolerance to noise, which is quite equal between the groups. It is significantly different, but it has no clinical meaning. These differences are so small. We also measured the distortion products of otoacoustic emission, which is the sounds that are emitted by the ear in response to a stimuli, and they're quite similar between the groups. And here you have the auditory brainstem responses from the two systems. Here is the Charter from Otometrics, and here the Eclipse from Interacoustics. You can see the wave shape is very different. The shaded areas show the confidence interval. And of course, this is a little bit larger because there were fewer patients in this group. And then the question was, how much difference do we have in the amplitude of the wave 1 response, which corresponds to the cochlear response? Here, what is the amplitude of the wave 5, which is corresponding to the inferior colliculus? Are there differences in the latency, for instance? All of these were questions that have been attempted to be addressed in the field for many years, but always with quite low sample size and maybe poorer tinnitus definitions. And this is what we wanted to address here. And what Nicholas found, he made a stepwise linear regression model. He found that something that was constant between occasional and control groups when compared to constant tinnitus is that the wave 5 latency was consistently greater in the constant group, which suggested that the ABR wave 5 latency could be a good biomarker for tinnitus. Now, I must say here that these wave responses, and this is what is complicated, you can see here, for instance, the red, there is a lower amplitude here in the constant group. The wave 1 can be significantly impacted by sex, age, hearing loss. So these aspects needed to be addressed in that evaluation here. And the amplitude of the other waves could also be affected by hyperacusis. This has been another work by colleagues that are in Tübingen in Germany. They found that these waves could be impacted by hyperacusis. So when we made this analysis, we took into consideration age, sex, hearing loss, hyperacusis, but also the hardware to make an analysis and really define what was impacted in that group. But what's the conclusion from that analysis is that constant tinnitus can be defined by means of the auditory brainstem responses, and thus we believe that it is a subtype that is really distinct from the occasional group, and that's the first way of classifying tinnitus. So based on that information, we conducted another study, which then tried to assess whether we could identify plasma biomarkers in the blood from people with constant tinnitus. And for this, we took the same cohort we have. Stop. Sorry. It's a little bit dry here. And we took plasma from 512 people that had constant tinnitus and 512 that had no tinnitus. And we analyzed 192 proteins from the O-link system. The O-link is described here. It's an immunoassay in which the antibodies are tagged with different primers. These are extended by PCR, and then you can read the amplification or the normalized level of expression corresponding to the levels of the proteins in the blood. This system is called proximity elongation assay, and this is a system that has been invented in Uppsala, where Kia has been working during the 2020. So we took this and submitted it to the analysis, and we made a very dense and really secured, I'd say, control for different factors: sex, age, BMI, smoking, and sample labs we found were influencing the data quite importantly, but also hearing loss. And with this, we found a number of markers that were just at the limit of significance here after correction for multiple comparisons, FGF21, MCP4. These were really interesting to see. And before we could tell that this was really a positive finding, we went for another cohort, which is the UK twins, and we performed the same analysis there on relatively the same number of samples, but we found no correlation at all between the two. So meaning that finally we found no proteins in the blood that could be associated with constant tinnitus. So the overall thought is that either we didn't have severe enough cases in our population to reveal something in the blood, or eventually we should have gone for cerebrospinal fluids to see proteins in much more concentrated forms that would allow us to distinguish between constant and no tinnitus individuals. So this is work for the future. Finally, knowing this, we performed a whole genome sequencing on severe tinnitus cases. And here the idea was to do the complete opposite from a GWAS. And this was, GWAS is large populations with maybe less defined and less well-defined phenotypes. Here we went for a smaller number of cases that would be analyzed by a very precise technology, and that's the whole genome sequencing. But we would go for a very extreme phenotyping. We would aim for people that would have severe and catastrophic tinnitus, chronic that would be constant and bilateral. So this type of phenotyping is really rare. So that's why it also explains the number of cases that we had there. But very luckily, so it was Sana Amanat has been working on this, and she's found a number of relations I will show you afterwards. Our idea is to then go back to the animal models, check where those genes would be expressed, whether in the brain or in the cochlea, perform gain and loss of function studies in vitro if we can, and then go back to the animal models and assess their contribution to tinnitus. So what Sana Manat from the lab of Jose Antonio Lopez Escames did, she made a first exploration in patients that had Meniere's disease and with or without catastrophic tinnitus. And she found a number of variants that were associated with this trait. And this we were able to validate in our stop cohort with 37 participants that had severe tinnitus. In the genes that are mentioned here, it's ACAP9, TSC2, and ANC2, which are genes that are responsible in neurotransmission. They're quite broadly expressed in the brain, but the odds and the association were quite strong for these rare variants. So this at least was also a first start, complementing the findings from Clifford et al. On their genes that they found associated with tinnitus. Our theory for the evolution of tinnitus is that we would have low variants, those that were found by Clifford, and rare variants, those that were found by us with Sana and the group of Lopez Escamas, these would be predisposing genes. And there with time, we would have a number of events in life in the environment that could be hearing loss, occupational noise, TMJ disorders, otitis media, and head injuries that would increase the risk of having first occasional tinnitus, that means with relapse and remission. Then some unknown factors would make that transition to constant tinnitus, where we would have ABRs, wave 5 latency changes that would make it much less likely to remit. And afterwards, there would be components involved in the severity of tinnitus through the combination of the auditory pathway syncing with other brain regions, such as the limbic system, and we would have the contribution of somatic input, cognitive thoughts also that would increase this clinical threshold to something much more severe. So this is now something that we would like to investigate in the future and try to understand how the different combination of factors with their interaction with genes contribute to first the development of constant tinnitus and chronicity, as well as the severity. So the conclusions for this talk is that we believe that a good definition for tinnitus should be chronic, constant, severe, and bilateral, which appears to be one of the most reliable traits for biomarker discovery. But one of the caveats for this is that this type of phenotype is a very, very rare condition. So the types of cohorts we have wouldn't even be big enough. So there's a big need for biobanking efforts across different countries. So this is why this is also a call for people to start collecting data in a standardized way and to centralize the efforts for sequencing, as it has been done by the Psychiatric Genomic Consortium, who's been doing a lot of work on all the psychiatric disorders that you may have eventually read throughout the Nature Genetics publications they have. We see also that the current biobanks have definitions that are too broad and unspecific, and even the definitions of the ICD codes that have been established by the WHO. So we have a number of biobanks that have ICD codes. They show very disparate prevalence in different countries. So for instance, Finland and Sweden have a prevalence of 2% of people that have severe enough tinnitus to get a diagnosis by a doctor. But in Estonia, this raises up to 20%, which means that Estonia has a different way of taking care of their tinnitus patients, and this could also be an issue. So not only we need a standardization in the research, but also in the clinic. For genetic biomarkers to provide robust output, this is really fantastic. But we see that plasma is much more subject to variability and sampling errors. That's why I think that the future biobanking efforts should focus on the genetic biomarkers. And while this is appropriate, in large consortiums, there is a good recommendation we provide here for ENT clinics to start collecting blood, but also cerebrospinal fluids and DNA, and this should be becoming a standard practice in ENT clinics. Here is my small lab, Niklas Edvall and Natalia Jabshevska, who are PhD students on the way. Evie just recently graduated. We've been working with different consortiums that have helped me stepping from the mouse models and jumping into human research. We've been also partnered by different pharma companies. We had funding from Sweden, but also from the British Tinnitus Association. I was shortly supported by the NIHR in Nottingham before my transition to Switzerland. And then we had European grants from the Marie Curie and the GenderNet to address gender issues in tinnitus research. I'd like to give some special acknowledgements to Barbara, who's been a fantastic mentor through my time in Sweden, and Jose Antonio, who's been also a fantastic help for the analysis in genetics. Winfried Schlee, Francis Williams, Silvano Gallus, Birgit Mazurek, Bertolt Langut, and Jan Bulla have been very long and fidel collaborators throughout this time. And I would like also to thank all the different big cohorts that have helped us doing this research. With this, I'd like to thank you, and I guess we're up to questions.
Thank you very much. Fantastic. So interesting to see the whole genetic part of tinnitus. Yes.
It's been a hard work for tinnitus,
yes,
really.
I truly believe you. But it's so interesting to see how it's moving forward. Yeah, we have hands clapping, no sound on. But yes. So open for questions from the platea. Cecilia, also a professor from Rio de Janeiro.
Hi, Christopher. Thanks. Great talk. I was wondering if you have quantified cortisol or inflammatory patterns in these cases of constant tinnitus.
Yeah, so all the 192 proteins that we assessed with the O-link system were from the inflammatory panels and also the neurology. So that we did check, and none of the inflammation appeared there. But that's why we believe, there's been evidence from rodent studies that there was an involvement of TNF-alpha in the auditory cortex. So this was one of the triggers for our analysis. And this is why I think maybe in the blood, it's not good enough. We have several people at Karolinska working with chronic pain, and they figure out working with the plasma is not specific enough. So they went into CSF, and there they could find an amazing amount of different biomarkers for chronic pain. The thing is that CSF is very delicate for collection. People need to be really voluntary for, getting a needle injected in their back. And I'd say not many countries allow this in terms of risks, but Sweden does. So maybe there is an opportunity to work with people and the doctors at the hospital and try to get this collected in an appropriate way. We didn't go for cortisol, and there are many reasons for this. It's complicated, it's pulsatile, it's circadian, so there are multiple factors we have to control for when checking cortisol. I know there were colleagues in Nottingham trying to do that from saliva, and I don't know how reliable this could really be. Now, we've been working ourselves with cortisol in mice, and really there is an immense impact of the genetic background into how the patterns grow over time. We see also in different rats that some animals have very pulsatile, ultra dial, we call that, cortisol profiles, that makes it very difficult to compare across groups. So I don't even know if a sample size of 500 people would be sufficient to get the resolution and the power for such analysis. But there is definitely something that has to be done with this in the future, because the links between stress and tinnitus are not strongly established now. There is a feeling for that, but the evidence is not strong, and it would definitely come with biomarkers like cortisol.
What if you give the rats cortisol or corticosterone, which would be the rodent counterpart?
So for tinnitus, we don't know, and for hearing loss, neither. But what we found with regards to circadian rhythms, we found that if you expose an animal in the morning and you expose the animals at night, there's different outcomes. So when you expose the animal in the morning with a certain sound level, their hearing is shifted for 24 hours, but then it comes back to normal. So it recovers. So the ear has an ability to restore the plasticity and restore hearing. But that doesn't happen at night. And we figured that was probably a mechanism related to the brain neurotrophic factor. And this is strongly expressed during daytime after a noise damage, but not at night. So you can rescue hearing if you inject the neurotrophins at night. So that works good. Now, strangely, when we remove the adrenal glands from the animals, we lose that day-night noise difference in noise sensitivity. So there is an involvement of corticosterone in the process, but it's against our assumptions, because we would think that the rays of corticosterone in the rodents at nighttime would be protective, but it's not. It's only when it's suppressed that suddenly it becomes protective. So what we think is that the involvement of these hormones is dependent on the time of the day. So probably you would need to give corticosterone during daytime to increase protection, but you would need to block the effects at nighttime. So in rodents, that you shift the activity time. During the active time of the rodents, you need to block the action of the corticosterone there, which is something we haven't tested yet, but is definitely something that will have to be done in the future.
We give corticosterone in drinking water for the rats and study depression. So that was it. Thank you.
Thank you.
Great. So then we have my colleague, Professor Tarciso. Next question.
Hi, Christopher. Thanks for the great talk. I actually have a question about the progression from sometimes to more frequent. How fast is this process, and does age affect it at all?
And what's the last question?
The age of the patients, is it faster in younger or older people?
Yeah, we didn't have enough samples to break it down in different age groups, so that is hard to answer. Now, tinnitus is definitely much more prevalent in the older group. So there, I'd guess, when you check at the demographics of the population, I guess it's the older group that was assessed. It's rarer in the younger ones, so it's hard to say. Now, in terms of timing and how fast the transition is, it's hard to say, because when you ask people, do you have tinnitus occasionally, even some people can say, well, sometimes I have just a beep that lasts 2 seconds, or it can last over a day. And even in there is a problem in how we define what is occasional tinnitus. But there's been a study performed by Dave Bagley from Nottingham that's been using the UK Biobank data that had been collected after several rounds like that too. And they had a little bit more nuances than we had. Unlike us, the UK Biobank data doesn't have a good definition for what is constant tinnitus. But what they could see is that progression from occasional tinnitus, it's highly dynamic. But it's very hard to say then, it revealed that people could have a recall bias there too. So when you answered, you said, well, I got sometimes a little buzz beep like that, and then it vanishes. But 2 years later, when you ask the same people the question, they can say, well, I never had tinnitus in my life, because that occasional tinnitus was so irrelevant that they forgot about it. Now the transition with constant, I think that we should focus on groups that have been eventually exposed to environmental noise. For instance, they could have a better recall for what they had before, or people in the music industry also, young people that have gone to concerts and discotheques, they have a buzz that lasted more than a day. And these would be a better target group to understand how this evolves through time and what could be a potential trigger for that transition to constant. My belief is that, and that's my own hypothesis, it's not, I have no evidence to claim this, but I think that there would be like the ear has a capital for damage. And at some points, once you reach your threshold, this is when the transition happens. You see the same thing happening with diabetes. People eat too much, people get obese, people have hyperinsulinemia. And then suddenly when they reach a threshold where the pancreas cannot tolerate the level of glucose anymore, then there is a collapse. There is autoinflammation going on, and then nothing works, and diabetes has developed. And I think that for tinnitus, something similar can happen, still being dependent on the periphery. So I think there is a relationship with the ear being damaged, maybe too much, or in a way that suddenly there is a cutoff and tinnitus becomes permanent. But how to address this experimentally is a bit of a challenge.
So I have a question or a comment. I think it's so important that you, as you were saying, that you've been interviewed a lot and you're in the media and you're trying to highlight all these things, because just this thing that you were showing now, that hearing loss, that you saw genetic differences in the stria vascularis, you know, that it wasn't necessarily the inner hair cells that were damaged and these things. I think it feels like it's so much old, I was almost going to say urban myths in tinnitus, that people have learned that, yeah, you damage your ear and there's nothing you can do about it. So it's not even worth going to the doctor. So I think we lose a lot of possible patients, possible blood samples and so on, just because there's such a negative stigma to the tinnitus. And even in Sweden, people go to the doctor and it's just like, yeah, you just have to learn to live with it, you know? And I think it's very important now that we highlight it, because I have people writing to me here in Brazil also, and they come with the most old-fashioned ideas of like they're wearing ear protection. It's just like they're so afraid of noise, and they know like if I have tinnitus, I'm going to go deaf in 10 years. I'm like, there's not any study saying that, you know. And then they have found it in an old textbook that is like 15 years old or something, and you're just like, so how do you think that like we need more media attention, right? Just to be able to extract the data to say what it is, right? So I guess my question is not a clean question, but do you see differences in different countries for this? Because Sweden is a country where you shouldn't go to the doctor, for example, but they have a lot of biobanks. And then you said in Estonia, they were more likely to take people serious. Here in Brazil, we have, there's the private hospitals, you know, so people go to the doctor for everything because they want to have their insurance money worth for something, you know. So it's very interesting.
I mean, the heterogeneity of tinnitus is followed by the heterogeneity of the healthcare systems across the world, you know.
Yeah.
And there was another student from Nottingham, Roshni Biswas. She's working. Hopefully, it's soon published. She made a survey on tinnitus across different countries in Europe, and she could see a bit of the prevalences changing there. So, irrelevant of the healthcare practice, there is already a difference in prevalence. Now, in different countries, I'd say, you know, very few countries have guidelines on how you have to assess tinnitus. America has it, but America has it because they are aware that it's an important condition that veterans experience, you know, after war. So they have research programs to try to help people with this. And so as England and then the Netherlands, they have also very good guidelines for this. Germany has stepped in as well. And then there's not much more because people don't really know what to do and what to offer. So as you say, you know, even in Sweden, if there's not much to say, cognitive behavioral therapy is one of the standards. It helps coping with, but it doesn't give a cure. Now, what I see as a big challenge is that as soon as there is a small test that becomes, or a therapeutic intervention that is becoming eventually available, ENT people are not the ones that are used to the research, and they are the ones that would have to perform a research locally to convince, you know, their government that the tinnitus can be treated, tinnitus can be handled, so there is even more work to do behind that. That's what I meant in answering you, Katarina, is that to make the change happen, we need the ENT people to become researchers on top of their practice. And making that change of mind is difficult. You have other clinical areas like inflammation and cancer. They've been used to doing research for decades. But to make that shift happen into clinics that have always been used to just make, you know, an assessment and a treatment with hearing aids or cochlear implants, there's something else to be done. And this type of mentality change in the research is quite a hurdle jump to pass. But hopefully this will make a change too.
Yeah, no, that's an excellent suggestion. We should team up with the clinics.
Am I answering your question?
Yeah. No, it's. I mean, now we have a question from Marini.
Hello, Christopher. I'm very happy to listen your presentation, and thank you to Katarina for this moment. I have a curiosity because I study clinical research. We have a team here at the university in Paraíba, a volunteer team of professionals, and I would like to know if you know when the ENT should request the genetic evaluation for our patients.
For the patients with hearing loss or patients with tinnitus?
Tinnitus.
So I don't know. So I would say if tinnitus is a primary complaint, that it doesn't come like for hearing loss. People come sometimes to the ENT clinic because they have hearing loss, and they ask the question, "Do you have tinnitus too?" Yes, but it might not be bothersome. You want the tinnitus to become, the primary problem they come for. That's one thing. Then it has to be constant. It has to be chronic. That means you have it for more than 1 year, and then it has to be also severe. So if you have the typical questionnaire scores like the THI that have been translated in Portuguese, that is a very good, valid tool. But then that might not be sufficient. You want to make sure, does it run in the family? Do you have siblings that also have it? Do your parents have it too? So you need to draw a family map and check whether there is something that is hanging in the family there, if it runs in the family. And if you have all of that together, then that becomes a strong indicator of a genetic risk. And then you could potentially do some Sanger sequencing, checking for the variants that we found in our paper, for instance.
Thank you very much.
And now we have a question from a PhD student here at the Institute, Juliana.
Oi, hi. Sorry, I was turning off the volume on the other sound system in the house. Hi, thank you for your lecture. It was really interesting. As a neuroscience PhD student, it's a lot to new things to learn, and also as someone who have tinnitus, it's also good to know. In your lecture, you mentioned the structure used to understand psychiatric disorders as a model to be applied in the tinnitus research. Were you talking about the RDoc matrix?
No, I was referring to the Psychiatric Genomic Consortium, the PGC. It's been led by Patrick Sullivan. They've been working now for 10 years doing a lot of GWAS study. And they started really as a small group. And it's a first effort between different psychiatrists that together had schizophrenic patients. And each of them, isolated in their little clinic, started to send the DNA to Chapel Hill in the US, where everything got sequenced all at once. And that strategy was very clever to do that.
Yes, it seems really interesting. I already took note of it. And but I don't know if you're already if you are familiar with the RDoc matrix, because they have this. It's an NIH initiative, so maybe it's not big in Europe. I don't know, but I particularly find it interesting because they have this units and constructs to work with cognitive systems, and where they put auditory perception inside of it, and they have this proposed to work with genes, molecular to unify information about genes research, molecular circuits, physiology, behavior, and etc.
Excellent.
So I find it's been an interesting tool for me to in my work, but it's focused on psychiatric diseases, and then I started to disorders, and then I started thinking about how much tinnitus could be considered a psychiatric disorder.
Yes.
What's your opinion on that?
That's a very interesting question, Juliana, because recently we were a bunch of researchers trying to redefine some forms of tinnitus. And we had a paper about the tinnitus disorder. which got a lot of criticism, but I'm fine with that because we just wanted to start launching the debate about new definitions. And in that paper, Vanessa, who's a good tinnitus researcher, started, asking what would be the contribution of those traits that are typical of psychiatric disorders, major depression, super anxiety levels, and how could that be a particular phenotype within tinnitus too, sufficiently different that we name it in a different way. And we got a lot of oppositions from people, for instance, from New Zealand, because there, if suddenly tinnitus was categorized solely as a psychiatric disorder, people wouldn't be able to get a referral and being taken care of. For a reason I don't understand, New Zealand wouldn't take care of tinnitus if it were defined as a psychiatric disorder. So there again, we're talking with Kia before about how different it is between the countries, and it seems that that's why we need some kind of coordinated effort to stratify things. I believe some groups of people have a high load of psychiatric distress that it could be defined as such, but not tinnitus in general. You see, the problem is that we use the word tinnitus for all the types that are there available all the time. We talk about tinnitus if you had it 2 seconds, 1 day, or your lifetime, and that's wrong. When you go to the doctor because you have high blood pressure, we don't say you're hypertensive, but you need to go several times, 4 times in a row, at 2, 3, 4 weeks intervals to the doctor before he says, okay, you've had high blood pressure throughout that time, now we diagnose you with hypertension. And those type of definitions, that are done in consensus with the majority of the clinicians is really important to do in a similar way in tinnitus, but on top of that, to include the different layers. Now, for instance, there's been a work from this group in Tübingen I mentioned before that shown that tinnitus with hypertension is different than tinnitus, not hypertension, but hyperacusis is different electrophysiology from those that have tinnitus without hyperacusis. And that could be a first type of stratification. And then there are other people that have this stress, as you're saying that, but we cannot say that every tinnitus is like that. For instance, in our case, we have in our cohort 30% with chronic constant tinnitus have significant stress at a clinical level, but only 10% have depression from using a questionnaire that is standard in the clinical setting. So I would say that maybe 10% of all the chronic tinnitus and constant could be potentially defined with a psychiatric problem, but not all of them. So there's a need for a lot of nuances before we can do that. Is it answering your question, Juliana?
Yes, thank you. It was very elucidating.
Okay, great.
I just saw we have one more question. I'm just going to stick a question in between. Do you think, by the way, congratulations on your latest paper there, this wave 5 ABR wave, do you think that could be a measurement that you can take at repeated time points? And,
yes. Why? You wouldn't dare?
No, I don't. I'm saying, wouldn't that be perfect then if you can actually follow that, if your tinnitus is more severe or less severe, and you can actually get a graph where you can measure
yes.
that?
So
would be
in
very
fact,
interesting.
that is one of the next projects we want to do. We were thinking of using lidocaine. So lidocaine, this painkiller, has been found by the dentist to suppress tinnitus. So the first, that was 40 years ago, that was just surreptitious. So it wasn't on purpose. So people with tinnitus went to the dentist, got their injection, and suddenly the tinnitus was gone. And this was fantastic. So in the States, they were so happy they started to open clinics to start treating everybody with lidocaine. But then this effect was just transient. And then the issue is that while the tinnitus would come back, people were very depressed about it. And lidocaine you cannot give chronically. It has too many side effects. It's quite dangerous to do it like that. But still, first thing is it's a proof that tinnitus can be treated with a medication. That's one thing. It has been one of the projects from AstraZeneca over the last 10 years before they reorganized all their structure. They wanted to transform lidocaine to make it with less side effects and give it as a treatment option for tinnitus. But this didn't work then in the end. It was too complicated. But this can still be used as a model to test with the ABRs. If someone comes in with constant tinnitus, you treat them with lidocaine. Do you restore the latency? And once tinnitus comes back, is the latency restored, proper to the level of a definition for constant tinnitus? And that would be a very nice evidence that you can track an evolution and the degree and severity of the disease.
Great.
But we need funding for that.
Yeah. Now we have another question from another colleague of mine, Rodrigo.
You're muted, Rodrigo. I don't hear you.
Sorry.
There you go.
I had the mute. It was muted. Thank you for your talk. I have two questions. One is related to, do you see early onset cases of tinnitus? And if these cases were studied, and are those the genes that were related that you show in the table, TSC2 and ANC, if I'm not wrong? Because in other neurological disorders, you have that has the genetic and environmental etiologies. Usually you have cases that are more genetically related, and they have this early onset case that sometimes can help you to dissect the genetic, pathways involved in, I would say, a more clean case.
Yes.
Do you have these? And the second question has to do with the group of cells that you kind of show this. You had this whole bunch of cells that were separated, but you focus and you show. You think that all tinnitus cases, they funnel to these two types of cells?
So I'll answer the first question and the second one. For the first question, the age of onset, it's very interesting you talk about that, but unfortunately in the questionnaires we've been including, and we designed that in 2014, we didn't think about it. But this is something we want to do now, because clearly we want to see, the earlier onset and the less influence of the environment you would have on the trait. So definitely it would be a cleaner phenotype. But I would say this is even rarer than the one we are trying to look at. If you're already selecting chronic, constant, severe, and very early onset, this is very, very rare. So we don't know yet how to address that, with the exception of these large biobanking efforts, but everybody is working together to try to do that. It's definitely worth standardizing methods for the assessment, and verifying what we can do. So it's good you remind me of this. We were discussing with other people, what should be the key things to gather as information for a biobanking effort, but an age of onset should be there. So it's good you reminded me of that. With the other question of the cells, the ones I've showed you, it was based on the GWAS on hearing loss, not on tinnitus. So that's one difference. We want to do the same for tinnitus, to ask the question, what's the cell contributing to it? And there we have an issue in the power, because you saw the GWAS on tinnitus had very low signals that were positive, and we need much more cases and controls. We need to increase. I mean, we have 700 people, but we need better definitions and much more sample size to get signals that are sufficiently strong to test that enrichment in the different cells. At the moment, with what we have, we can't do it. We don't have enough statistical power. But we have to test it out at some stage. Yeah.
Right. Thank you.
Pleasure.
Could I make another quick question, Kia?
Sure, go.
No, just as a curiosity. I'm sorry, my English, it's a bit rusty.
It's very good.
Oh, thank you. You were talking now when answering to Rodrigo. I just remember about neurodivergent populations we are starting to get more information about ADHD genes and autism genes, and the combination of tinnitus and hyperacoustics in this kind of population. I believe that, not just saying, I'm guessing that it might be a phenomenon. Do you know something about it?
No, none, unfortunately. And that's another issue, is that with all the biobanks we have, there is absolutely no information about hyperacusis. These biobanks were created maybe 20 years ago when people started to talk about tinnitus, and that's why the simple question went in there. Have you ever perceived tinnitus once in your lifetime more than 5 minutes? And then that was just the worst question you could ever ask, because it's so undefined and unprecise that you cannot do anything with this. But then even hyperacusis was not even considered. Hyperacusis starts only now to become something that people want to start focusing their research on. And it starts now to become an important trait to include in all the tinnitus studies, also as a covariate and a factor that you need to adjust your analysis on. But, definitely the combinations of the two would have to be distinguished. The people believe that the hyperacusis would be related to the increased gain that you have in the brain as a compensatory response to hearing loss, and people believe that tinnitus instead is not that increased gain, but it would be more a bruisey signal that emerges from that noise. So the two would be a bit separate. A lot of people that have tinnitus also experience hyperacusis, but it's not always independent. What we think is that tinnitus could emerge, and in particular in women, first, and could be like a predecessor to the development of tinnitus. But this is something we need to test as well.
Thank you.
So, do we have any final question? If not, oh yes, we have. Rodrigo.
May I just a quick question that I was just thinking about the early onset thing is for diagnosing tinnitus in children. Is there any test that is as objective as possible to detect tinnitus in a population that sometimes cannot express the feeling of this noise coming in?
I mean, one could then think about the ABRs, right? But I think the complexity here is that if you want to transpose such a diagnostic in the clinic, you need eventually machine learning, because just out of the raw data, you cannot make any extrapolation, because it's so much influenced by your sex, by your age, by your degree of hearing damage and hyperacusis, that all, the amplitude and the latency will be impacted by that. So you need to integrate in your clinical setup some kind of machine learning that says, all right, now with your parameters, we expect a likelihood of 90% for you to have constant tinnitus. I think that could be the future, and I guess that one could apply it also to children. What I think would be a very challenging aspect is to do it with people with autism, because they have high vulnerability to loud sounds. And typically with ABRs, you need to expose people to a level of 80 decibel hearing levels to get a signal, and that could be a population that could not benefit from such diagnostic.
And now we had a question popping up from George also, another colleague here.
Yeah, thanks for the talk. I appreciate it. Thank you very much. So my question is not really related to the talk you gave, but I'm curious about your animal tinnitus model. Can you talk about it?
Yes, I can do that. The animal model, I might have the kids coming in a very few seconds. So if it rings, then I'll isolate myself in the room, but just don't worry about that. It's just a big tribe.
Well, I'm just gonna go for another 5 minutes, then you're free. So
it's really fine. They understand that, but it's the timing where it starts ringing in the bell door. The way we started working with the animals, so that was one of my first work as a postdoc. I've been working with mice, and I thought it was really a nice model to have all the genetics that was available to test, what is the function of a gene. You remove it, and then you can assess the phenotype. And one of the first questions is, how do you assess that in animal models? And when I tried with rodents, all the conditional behavioral assays that were very challenging in mice, but maybe I simply missed the skills to do it. So I went for a more objective measures like the startle response. And there is a phenomenon called the gap detection prepulse inhibition of the startle response, which is influenced and inspired probably of the prepulse inhibition that has been used in schizophrenia, in which for prepulse inhibition, you have just a small of sound before you have a big startled pulse that, you know, the animal jumps on a platform. So if the animal perceives that prepulse well enough, the startle response is inhibited. It's like a pre-warning system that prevents the animal from startling. And for tinnitus, you use the same kind of principle, but instead of having a small prepulse that precedes the startle, you have a continuous background noise, and then you have a silent gap, and then it continues. And you can narrow that carrier noise within different frequency range, like low frequencies and other frequency levels. And then you can test out, you know, is tinnitus impairing the ability of that silent gap to inhibit the startle response? So when we started with animals, I wanted to use salicylate as a model, which is quite robustly triggering tinnitus in rats. I didn't want to use noise because often 50% of the animals developed tinnitus and 50% did not. So if I wanted to test the reliability of the startle system, I needed something more robust. So we went for aspirin, salicylate. And then in wild-type animals, there was such a resistance to that. I mean, the changes were minimal. You were having a very good inhibition at baseline, and then it would decrease by maybe 10%. And this I couldn't really trust. That for me wasn't like an effect, could have been really random. In fact, people working on schizophrenia with prepulse inhibition have published, you know, effects like that. Oh, we saw a change like this. The animal is schizophrenic, and no, I wouldn't trust on that. So then I started reflecting on why would mice be maybe more resilient to develop tinnitus than the other rodents, where guinea pigs and rats have been traditionally used in the field in the last decade. And I figured out that in the ear, there is a glutamate transporter that is very abundant in the cochlea from mice, but not, it's very hardly detectable in guinea pigs and in rats. And I thought this could maybe be a reason. So the glass transporter is present on the pillar cells, the inner phalangeal cells, and underneath the inner hair cells, and they suck up glutamate when it's released in excess by the inner hair cells. And if glutamate is too abundant, then you have excitotoxicity at the level of the spiral ganglion neurons, and this could potentially, you know, trigger something higher level in the brains and potentially tinnitus. So when I took mice that were lacking that glutamate transporter, so they had no ability to buffer glutamate whatsoever, and I injected them with aspirin, then I could see the impact on their gap detection levels were really dramatic. And this was narrowed down at a single frequency, which was 32 kilohertz. Broadband inhibition was really not affected. Prepulse inhibition was equally similar between the knockouts and the wild type. So we were kind of confident that we were able to trigger tinnitus in that animal model. Now, it has been quite difficult to work with it because the animals that lack glass, 50% of them die at birth. So it complicates the breeding quite a lot. We need to develop some fluxed animals and cross them with specific Cree lines that would be targeting the support cells in the cochlea to make it much more effective. So that's the stage we're at.
Okay, thank you.
So, if there's no further question, I would like to thank you so much, Christopher, for talking to us here in Natal, or also in Rio de Janeiro. We had one person, and we're gonna, and yes, in João Pessoa, Marini, of course. And we're gonna upload this on our YouTube channel. So if you have students from your groups that want to watch this talk again, I'm gonna send the links. No, but there was lots of information here and lots of interesting things. So thank you so much.
And I'd like to say, if the students would like to contact me personally, if they want to continue the discussion for whatever aspects, whether it's, you know, scientific or career-wise, I don't mind. have traveled enough and done sufficient things wrong that I can give good advices what to do.
Yeah, no, we usually actually have a seminar series about on career steps. So maybe we can invite you in the future for that too. Like what to do and what to not to do. But your career is fantastic. So anyway, thank you so much. And I hope in the future, when the pandemic is over, that we can one day invite you to come to actually come to our institute and be part of our house symposium and come to the beach and have a drink afterwards and everything.
I'd be very happy to do so.
Looking forward to that day. But for today, thank you so much.
My pleasure. Thanks, Kia. Thanks to everybody.
Yes. Bye-bye. Have a good lunch,
Bye-bye.
everybody. Ciao, ciao.
Bye, Kia.
Bye.
Bye. Thank you.